Hello, my name is Josh, and I'm a junior biology education major.
And I'm Hannah, and I'm a sophomore chem major. And today we're presenting our research on the investigation of the release and uptake of dyes and pharmaceuticals from chitosan alginate bioplastics that we did in Dr. Wiley's lab over the summer of 2021.
Yeah, so where our research really came out from was the general chemistry laboratory at Concordia College. The Gen Chem is pretty much a course that most first year students take in the sciences. And part of that course, Dr. Wiley has incorporated a course-based undergraduate research with these chitosan alginate bioplastics. And we really wanted to investigate everything about these bioplastics and gain a just better understanding of how they work and how they could be used further in the lab going on in the future. So what these chitosan alginate bioplastics are made of is chitosan alginate, two biological materials out in the world that can be used as a sort of sustainable version of plastics. And chitosan comes from the shells of crustaceans and fungi. Chitosan can bind to itself in very long chains, which makes it good for producing films, but on its own, it's not very strong. So that's why we wanted to introduce it to alginate. Alginate is found in algae, and like chitosan, it also can make very long strands with itself. But the power of this bioplastic comes from when they interact together, creating cross-linking. So we have chitosan-chitosan interactions, alginate-alginate interactions, and then chitosan-alginate interactions. And this cross-linking creates a matrix that creates a very flexible plastic that also allows us to embed things into the plastic itself, which is where really a lot of our research comes from.
So like Josh said, we can embed things into these bioplastics. And the first few things we chose to put in our bioplastics were food dyes because they were readily available in the lab and they're really easy to use and easy to see. As you can see on the slide right here, just note that these bioplastics, these food colors in these bioplastics, though they seem like similar things because they're all dyes, their chemical structures are not the same. They're actually very different from one another.
Yeah, so release was kind of the very first thing that we looked at in our experiments, because we can put this food dye into the bioplastic, and we wanted to see how much would come out in a release system. So one of our basic experiment conditions would be is that we would have a set solution volume, either 10 or 20 milliliters. We would put a standard mass of bioplastic into them, and we'd just simply measure how much color would come out of the bioplastics over a period of time. And these food dyes, they're obviously not all the same, so they're going to absorb and reflect light at different wavelengths. So we have those wavelengths up there, which is where we measured their absorbances from, or how strong of a color we were seeing in the surrounding solution. And so over time, we would see obviously an increase in how much dye was coming out of the bioplastic into the solution. And from the graph, you can see that there's a large initial release. Then over time, it sort of plateaus off. And 200 hours was where we found our equilibrium point to be. And just initially, we wanted to try different solution environments. So we chose RO water, which is reverse osmosis water, which is a type of distilled water, and then a salt solution of NaCl. And we could clearly see that the salt caused a much larger release. And so we wanted to scale this up. This was our initial test with just blue one. And we were thinking, oh, that should behave the same similarly for the rest of the food dyes. But we saw that it actually didn't. Here I will mention that blue 2 and red 40 are some of our problem children. They didn't behave as we expected them to. So we'll just take those off our graph and we'll see the four most well-behaved ones. As you can see, there's an order here. Yellow 6 releases the least, while yellow 5 releases the most, and blue 1 and red 3 are in the middle. And so they're not all behaving the same. And they also don't behave how we would expect them to when we put them in salt. So here's the graph of all the four most well-behaved ones in salt. And as you can see, the order has changed, it's shifted. And two of our colors went off scale right away. So there's something else going on here. The relationship between the solution environment and how much dye being released is not as simple as we originally thought.
So we decided to look at the reverse of release, which is food dye uptake. And for uptake, our systems had strips of colorless bioplastic with nothing in them. So we dubbed them vanilla bioplastics. So there is no dyes or pharmaceuticals added to these bioplastics. And we put them in 20 mils of dyed water. So it would be our colors or our pharmaceuticals in these 20 ml vials with this vanilla bioplastic. And you can see some of our systems here. And for uptake, our graphs read like the reverse of release graphs. So they start off high and they go low. And if we see this decrease in the concentration of, in the absorbance of these dye systems, we know that dye is getting uptaken into these bioplastics. And you can see here are our 4 well-behaved dyes, and we see that, yes, dye got uptaken into these bioplastics from the solution. But if we look at these in salt, we see again, the order completely changes. Red 3 uptakes a lot, but the rest of them don't seem to, and that we will explain later. But also we left off 2 very important dyes off these graphs, and that is for a specific reason. So red 40 and blue 2, these are our problem children of all the 6 food dyes. They just don't behave how we would expect them to in release nor in uptake. So you can see from this photo here that there's something a little strange in these vials. There's these weird freckles in our solutions. And this is a strange precipitate that we were seeing in our red 40 and blue 2 uptake systems. So this happened in blue 2, but we have good photos for red 40 to showcase these weird happenings. So for release in red 40, we saw almost no release. You can see that these colors in these vials are very clear, not very saturated at all. And then in the beaker here is an uptake system. And there it is again, is a strange precipitate that we have no idea what it is. So we decided to analyze it, and we took it to the IR, and we compared red 40, just the dye itself, the pure red 40 dye powder, and this weird precipitate we were finding in our solution. And we can see that these 2 graphs do not exactly match. And that can tell us that these two materials are not the same. So this strange precipitate in our solution is not just red 40 dye coming out of solution. So we hypothesized that it could be some interaction between the chitosan and the red 40 itself.
Absolutely. So now we're going to a little summary of our food dyes so far. So we talked about that the orders aren't the same depending on the solution environment in release. We have a changing of how much gets released depending on if it's an RO, water, or salt. Then in uptake, we can see that red 40 releases the least in our uptake in the RO water, but then when we put it in salt, it's one that uptakes the most, and the rest of them don't uptake very much at all. So there's something interesting going on here. But generally speaking, if we see a large release, we get a very poor uptake, and vice versa. For example, yellow 5 releases the most, but then looking at uptake, it doesn't really uptake very well. And the same can be said, but in the reverse for red 3. So that's sort of a general observation that we made with our colors. And next part we wanted to tackle was why. Why are we seeing this sort of relationship? What can help us understand this?
So that took us to the chemical structures of these materials. And our first question was, what's with all of the release in blue 1 and yellow 5? And we think that is because of the charged groups on these molecules. So you can see that for blue 1 and yellow 5, they have 3 ions attached to them. So they're triionic salts. So that increases the amount of interactions and bondings they can do with our bioplastics and with the salt solutions in water. So that's why we think that they are stronger and better releasing than some of the other molecules. And as for red 3 and how it really likes to stay in the bioplastic in uptake and release. So in release, it doesn't release very much. And uptake, it really goes into the bioplastic a lot because it likes to be in the bioplastic. We think it's because it is a more non-polar dye. And that can be backed up by findings that were done by previous students in the Wiley lab. But overall, we can see that these are complex organic structures and have the potential for a lot of possible interactions that we need to investigate more of to figure out exactly what is happening between our bioplastics and these dyes.
So we also tested a couple of other materials in our lab. Once we had gotten a good grasp on food dyes, we wanted to see what else we could try. And one of those things was tie-dye. Our lab works a lot with tie-dye for other reasons in our science outreach program. So we had tie-dyes laying around in the lab, so we wanted to see how they would work. So we put tie-dye into these bioplastics and also into solution with plain bioplastics and see how the release and uptake worked for these. And tie-dye, being as it wants to stay enclosed, that's its whole point. And that's kind of what we saw with our release uptake system. So In release, it kind of wanted to stay in the bioplastic, which is much non-polar compared to the surrounding environment. And then in uptake, they generally wanted to go into the bioplastic as opposed to staying in solution, which makes sense for the purpose of tie-dye. In addition to tie-dye, we also wanted to try something else that was colored, which would be indicators. So pH indicators are often colored so they can be easily show the pH of certain solutions. So we wanted to test that. And this is very, very preliminary work. And obviously the bioplastic itself changes the pH of the solution that it's in. So it's very hard to kind of quantify the release that we're seeing because the color changes. That's the whole point of pH indicators. But it was still very interesting to see another route that potential students might take in the cure of the Wiley lab. And then finally, we also wanted to look at some pharmaceuticals. And previously in the Wiley lab, we've had students look at sulfa drugs. So we thought maybe putting sulfa drugs in the bioplastic would be a good idea. And so we did. We impregnated the bioplastics with sulfa drugs, and then had them sit in vials of release over a period of time to see if we could actually get some initial release out of these bioplastics. And we did. So this kind of showcases our bioplastics as a sort of potential drug delivery system, which again is another route this research could take with first-year students.
So to sum up some of our findings, one of our main findings was that these bioplastics can be very useful for a lot of different things: pharmaceutical release, uptaking of dyes, and such. And most of our work did focus on the dyes, but this allowed us to understand these systems to a very high degree when compared to what we knew beforehand. And our main takeaway was, if it releases a lot, it's likely to uptake less, and vice versa. So if it uptakes a lot, it's likely to release less. And that was one of our main takeaways. And we have been trying to coordinate that with molecular structure today as well. And we think that our research is most exciting because it allows students to have pathways that they can take inspiration from for these undergraduate research experiences in the Wiley lab. And it also allows for, more research from Wiley students that aren't in these CURE programs to really delve into a specific topic like we did with food dyes and these bioplastics. And we'd like to thank a few people, obviously Dr. Wiley for letting us be in his lab and choosing us to do research with him, Roger Egan and Analytical Instruments for providing our HPLC machine that we did a lot of our analysis on. So thank you, Roger. Dr. Strand and Erska for providing a really great program this summer for supporting our research and letting us learn about professional development and such, and Centennial Scholars for our funding, and the chemistry department for the lab space and all materials involved. Thank you.
Thank you.